Hi, everyone. Thank you all for tuning in today. Welcome to Conservation Conversations. My name is Ed Pritchard, and I work for Miami Eco Adventures. This webinar is a joint effort between the UF IFAS Extension, Florida Sea Grant, and my organization. We will be offering this webinar series every Friday at 1 p.m. and 5 p.m. for the foreseeable future. Although we can't see you in person right now, we are excited to offer this webinar series and reach even more people than we'd normally be able to in one classroom. So thanks so much for tuning in today. Everyone in this webinar is currently muted, so I ask that you guys type any questions that you may have throughout the presentation into the chat box, which I will moderate throughout, and we will answer the questions at the end of the session. Just a reminder that this webinar is being recorded, and a link to the recording will be sent out by the next day. Please follow us on social media, where we will be announcing each week's conservation topic at the beginning of the week. If you'd like to receive an email reminder with these conservation topics and registration links, please email Anna. She will provide her email or let us know in the chat box, and we will take note of that. So without further ado, I'm going to turn it over to today's presenter, Anna Zangroniz.
All right, thank you, Ed. Good afternoon, everyone, and thanks for being here. That was a great introduction, and this is me. I'm Anna, and I'm the Florida Sea Grant Extension Agent here in Miami-Dade County, and I work for the University of Florida IFAS Extension Service and Miami-Dade County. And we've been doing these webinars for about 7 weeks now since the COVID-19 restrictions began. So we're really happy to have you all here and to talk about different topics that are local and relevant to Florida. Before I get too deep into today's topic, I wanted to take you back to where my particular obsession with stingrays started. Some of you may have heard of a place called Stingray City. Stingray City is located off of Grand Cayman, and it's a place where stingrays have been fed by humans for about the last 20 or 30 years. And this has some controversy associated to it, but there are quite a lot of tourists who go to this site simply to interact with semi-habituated stingrays. And back in May of 2007, I was one of them. I was already an avid diver at this time, and nothing intrigued me more than getting the opportunity to get into the water and interact up close and personal with stingrays. And you can see that I'm holding a film waterproof camera, so some of you guys will appreciate the days of those. I don't even think you can buy them anymore. And I'm holding in my arms a giant southern stingray, and it felt like wet marshmallows, and it was just unbelievable. I later returned to Grand Cayman that summer for a full-fledged dive trip, and I experienced this site on scuba, and it was fabulous as well. And this led me, upon moving back home to Maryland, to become a volunteer scuba diver at the National Aquarium in Baltimore. And in this role, I was really fortunate to be able to feed all of the animals that lived in the Wings in the Water exhibit, which was one of the largest exhibits at the aquarium. And it featured seven species of stingrays, different sharks, and of course, Calypso, the 500-pound, 3-flippered green sea turtle. And these are photos of me from an underwater viewing window during a family night event. Behind me, we have a southern stingray. The ray that's flying by my head is a cownose ray. And then in the photo on the right is a rough-tailed stingray. And I became very close friends with all of these, and it only continued to pique my interest as to these species, because as a volunteer for the aquarium, we not only had to learn about every single species that we were responsible for caring for, but also all of the other creatures in the aquarium. So I became a semi-stingray expert, as well as telling you why to close your mouth when you looked up in the tropical rainforest exhibit. Now, my obsession with stingrays continued once I came back and moved to Florida for graduate school, and I started diving more regularly here. And the spotted eagle ray, or Aetobatus narinari, is featured here, pretty well done by Disney Pixar, and they did a really good job capturing what this species looks like. And for those of you who might be curious, The genus name of Aetobatus is derived from the Greek aetos, meaning eagle, and batis, meaning ray. And Newby et al. Identified one of the largest brain-body ratios in this species, largest brain-body ratios for fish, indicating that they're pretty intelligent. They can live for about 25 years. And physically, they're defined, they're pretty distinctive with this dark body and beautiful white speckle and spot patterns. They have the very pointed dorsal fins, and their tails are usually 2 to 3 times their body length. I really like this photograph because it can show just how large the species can get. It gives a little bit more scale when you look at it with humans into the picture. So these fish can get to about 8 to 11 feet in length from tip to tip of their dorsal fins. I want to take it for a moment and just talk about basic biology when it comes to stingrays. And I'd like to ask you all to go ahead and type into the chat box as to which of these spotted eagle rays you think is the male and which you think is the female. And I'll have Ed read out the guesses. So just take your best guess, which is the male spotted eagle ray, which is the female spotted eagle ray.
So Joanna says the one on the left is a male, as does Marcia.
All right.
Terry says the same, the male on the left.
Okay. Not bad. You guys are correct.
Yeah.
And I'm pretty sure you're guessing that because of the presence of claspers, which are the reproductive organ that are located under the tail of the stingrays. That indicates the male stingray, whether it's a spotted eagle ray or another species. Nicely done. All right, I'm gonna ask you guys a really easy question. If you guys would please, Ed, if you could launch this poll, this is just one question. When Ed launches this poll, you all could just take your mouse and click on the answer that you think is correct. One question, what is the difference between bony and cartilaginous fish? A, bony fish are skinnier. B, bony fish have skeletons made of bone. C, cartilaginous fish have skeletons made of cartilage. And D, both B and C. And we'll give you guys just a few more minutes. Go with whatever comes to mind. Your first guess.
All right, we got about half.
All right. We'll give it about 30 more seconds. So you guys just go ahead and answer what you think is the right one.
All right, still waiting on a few more.
No problem. And guys, if you just answer, even if you don't get it right, this guarantees you a hug the next time that I see you in person, and it is safe to do so. All right, Ed, let's go ahead and wrap that.
Sure thing.
And actually, would you just go ahead and share the answer?
Sure.
Thank you. All right, for those of you who guessed, the answer is D, both B and C. Bony fish, as the name would indicate, reflects that bony fish have skeletons that are made of bone, same as ours, and cartilaginous fish have skeletons made of cartilage. Thank you for playing, guys. Now, the reason that we like to bring this up, if my slide would change, there it goes, is when we think about what makes a stingray different from other fish. So we start to look at the taxonomy of a stingray. We know that they're in the animal kingdom. The phylum Chordata is indicative that they are vertebrates, and their class of Chondrichthyes means cartilaginous fishes. And within the class of Chondrichthyes, they're in the subclass of elasmobranchs. And these include sharks, skates, rays, and sawfish. The taxonomy breaks down even further into the order, family, and then genus and species of Aetobatus narinari. I really like this diagram. I think it does a terrific job of giving a really simple overview to just the differences that we have in our stingrays here. So you can see within the order of Myliobatiforms, I'm going to give a local example or ones that you're likely to recognize of each of the families and the genus. A local example of Urobatis is the yellow stingray, which we see a lot in seagrass beds and also around coral reefs in Florida. Daciatus includes the southern stingray, which is very common. Myliobatiform genus includes the eagle ray or the bullnose eagle ray. We had 2 of these at the National Aquarium in Baltimore. They were my favorites in the exhibit. We have Rhinoptera, which includes the cownose rays. The manta genus, which of course includes the manta ray species. And last but not least, our Aetobatus, specifically the Aetobatus narinari, which is also, in addition to its really cool spot pattern, is pretty distinctive because of its very fleshy duck-billed and pointy snout. Now, these fish are studied most frequently by acoustic telemetry, and this is a technique in which an animal is implanted or receives a tag, has a tag attached to it, and that acts as a transmitter. And that transmitter sends out a signal, which is then picked up by receivers that are placed throughout different locations in the ocean. And this is really common, a common way to study sharks, rays, and other types of bony fish. And so this information collects this technique collects lots of information about the fish's movements. This map is reflective of the distribution of spotted eagle rays around the world, and it's a pretty wide range. They can live in tropical, subtropical, and even temperate waters to a certain degree, found on and around coral reefs and also in shallow estuarine areas. And there are at least 3 confirmed species, but it's thought that there are up to 7 that are different. And I'd like to mention that these species aren't distinguishable to the naked eye. In other words, you can only distinguish between the species by taking a genetic sample. So first we have the Aetobatus narinari, which is ours, which hangs out in the tropical Western Atlantic, Caribbean, and Gulf of Mexico area. Aetobatus laticeps, which hangs out in the Indo-Pacific region, and the Aetobatus ocellatus, which hangs out in the central, western, and eastern Pacific. I would just like to say that my disclaimer is that these animations aren't exactly true to scale, they're just designed to give you an idea. Zooming in on a more local level, Sellis et al. Found a seasonal migration pattern from the central west coast of Florida to the Northwest Caribbean during winter months. Now, we joke about birds flying south for the winter, but that seems to apply here, that these fish don't like when the water gets to a certain cold temperature, so at least this population that's been studied is moving south to where waters are warmer. Sellis et al. Also found evidence of gene flow between Mexico and Cuba and Florida and Cuba, suggesting that this species are capable of pretty large-scale movements. I'd like to ask the group, a couple of volunteers, to type into the chat box what they think a mesopredator is. We know that sharks are predators or apex predators. What do you think a mesopredator is? Ed, do we have any guesses?
No guesses yet.
Oh, no guesses. All right. Well,
we have one from Joanna. Prehistoric predators, yeah.
Prehistoric predators, not exactly.
Jeff says a lower level predator.
Yeah, lower level. In other words, a mid-level predator. So it eats things and things eat it. And the spotted eagle ray has a wide variety of food sources. They mostly consume benthic invertebrates, including gastropods, bivalves, polychaetes, and in Bahamas, Bermuda, and the Caribbean, they have been found to consume queen conch. So their diet differs a little bit according to geographic region. And this information was learned not only through dissection of specimens collected either through bycatch or collected in fisheries, but also through gastric lavage, which essentially is stomach pumping. And it's a process where the animal is kept alive and water is pumped through the gills so that it can continue to breathe throughout this process. It's very similar to approaches that are used during shark tagging, if anyone has ever been a part of one of those types of trips. As I mentioned before, the spotted eagle ray can reach that wingspan, tip to tip, about 8 to 11 feet, up to a maximum total length of 17 feet, and up to about 500 pounds, which is a pretty huge stingray to meet, especially underwater. They reach reproductive age a little bit later in life, around 4 to 6 years old, and they're ovoviviparous, which means they reproduce via internal fertilization and give live birth. However, they don't connect to their young through a placenta like most mammals. Instead, their embryos live off of energy obtained from yolk sacs, and only after the juveniles are able to survive on their own does the mother actually give birth. They're pretty small amounts of pups, however, small quantity that is, only 1 to 4, and right now the research has shown that they have about a 25-year lifespan. Spotted eagle rays show lots of different behaviors. We've seen them before in large flocks or schools, and they do that both for social reasons as well as large-scale movements. However, when they feed, they exhibit behavior that's more solitary or individual. They travel throughout the water column, and it's been found that they can travel in waters that are quite deep, about 900 feet deep. They also fly or travel over sand flats. But these species don't bury in the sand the way that whiptail species like the southern stingray do, but they can also jump out of the water when swimming close to the surface, and this is usually to escape predation or if they're being pursued. Just like other animal creatures that live in the ocean, the spotted eagle ray face a great number of threats to their survival from both a natural or biological standpoint, as well as an anthropogenic or human-caused standpoint. As I mentioned a couple slides ago, they don't reach reproductive age until they're a little bit older, and when they do reproduce, they reproduce in extremely small numbers. They also have, they face threats from natural occurrences like red tide, which can affect their food sources, and they are also victims of predation. It's a natural part of the chain of life. Silver tip and great hammerhead sharks are predators of the spotted eagle ray, and sharks have been reported to follow spotted eagle rays during the birthing season, feeding on newborn pups. Something I'd like to note that I found really fascinating was that during the 2017 to 2018 red tide blooms on the Gulf Coast, all of the rays that were tagged in the area exited Sarasota Bay as red tide levels reached medium, or about 100,000 cells per liter of water, which is a concentration known to have negative effects on small fishes, which is again going back to that thought that they're incredibly intelligent animals. From an anthropogenic or human-caused standpoint, these animals are overfished. They're not a target of commercial fisheries, however, but they are targeted in artisanal and sustenance fisheries in both Mexico and Cuba. And there aren't many regulations for the fishery in Mexico. They can fall victim to entanglement and bycatch issues, as well as boat strikes, and water quality issues can reduce to quality and less availability of their food, as can climate change. There are lots of conservation efforts currently underway for the spotted eagle ray. One of the first and earliest was the listing of the spotted eagle ray under the International Union for Conservation of Nature's Red List as a threatened species. And the IUCN was established in 1964. It's a global union of states, governmental agencies, and non-governmental organizations serving in a partnership that assesses the conservation status of species around the world. And the listing of spotted eagle ray took place in 2006. And you can see that it's listed as near threatened, but their overall population trend is decreasing. I really like using this as a resource, the IUCN Red List, because it's based on scientific information. Although it's the first one to say we need more information. Locally here in Florida, the spotted eagle ray is protected in Florida state waters under Florida Administrative Code with the purpose of increasing public awareness of the need for extensive conservation action. You can see it right here. Since the spotted eagle ray is listed, it cannot be harvested, possessed, landed, purchased, sold, or exchanged in Florida. Now, the data that goes into setting up some of these policy recommendations and regulations comes from research. And the two approaches to studying spotted eagle rays, the first include aerial surveys where photographs are taken from a plane in a certain area over a certain amount of time, and boat-based studies that can include a combination either of tagging or photo identification or both. Now, Ed, I'm going to ask you to pull up one more little poll for me, please.
Sure thing.
And I invite you all who are participating to go ahead and use your mouse and choose on which you think is the correct answer. Why might the markings of a spotted eagle ray be used for identification? A, the population of spotted eagle rays is so small that it's easy to ID them by their markings. B, the markings are unique to each animal, like fingerprints. C, they are easily visible from a drone. Or D, dorsal study is the scientific standard for stingray ID. So we'll give you guys a few more seconds. Go with your first guess.
We have 9 of 16.
All right. Give it about 10 more seconds, Ed, and then I'll ask you to share the results, please.
All right. Okay. There we go.
All right, so great job, everybody. The answer is B. The markings of the spotted eagle rays are unique to each animal, like fingerprints. And there are other studies that use photo ID to help track population. Manta rays actually have a similar pattern in that their bellies, their undersides are photographed for the same reason, because they have a set of markings that are unique to each animal. I'd like to touch upon the two main methods for studying these animals outside of aerial surveys. And just like any other study approach, they both have benefits and drawbacks. When we look at tags, tags give the great ability to track movement and migration, even on larger scales. However, in addition to being expensive, it is considered an invasive approach, and it could alter the behavior and potential survivorship of the animal. Tags can also get lost, and eventually the animal could just disappear altogether. Photo ID is considered a non-invasive approach because those fingerprints, if you will, act as a natural tag. And that can be really helpful for establishing population parameters. If the photo quality is sufficient, it's a 100% accurate approach. The limitations to this is that it is limited to shallow waters, both from when boating and bringing the animal onto the boat. That water needs to be shallow enough to lower a net and still retrieve it. Likewise, if you're having individuals in the water, either snorkeling or scuba diving to take pictures, you are limited by depth. Same applies for turbidity and water clarity. And if you're working in an area that has other predators nearby, it might not be the best thing to have people in the water. I want to focus for a moment on a couple of the photographic studies. Most of the work that's been done in this part of the world is focused in the Gulf of Mexico and Mexican Caribbean. And one of the first was by Cerruti Perea et al. In 2018. And they created the first catalog of photo identification of this species in the Mexican Caribbean region. And this was a really cool project in which they assembled almost 1,100 photographs in a 13-year period. And in addition to researchers and recreational divers, a local nonprofit organization called Blue Corps launched a citizen science project, and they helped assemble historical photos of spotted eagle rays, but also put people in the water to contribute to photography to add to this project. And so this paper had a spike in their data set in 2015 when Blue Corps started actively photographing. And this greatly increased the capacity of this project because about 60% of the total photographs came from Blue Corps. And as a result, almost 200 or 282 individual spotted eagle rays were identified. And they also discovered a certain amount of site fidelity at particular areas within Mexico. And you can see that they focused on using the pelvic fins for their identification. Another study that I'd like to highlight was by Gonzalez Ramos et al. In 2018. And in about a 1 year's time, they photographed 191 rays and were photographing, these were through catch and retrieval onto a boat. They photographed on 3 different regions of the fish. They used the head, they used the pelvic fins, and they used spots along the dorsal fins. And so in this study, they identified 132 individuals and 59 recaptures. In other words, 59 individuals that had been photographed before and then captured again. And so they determined that the dorsal surface of the pectoral fins was the most accurate place to run their photo analysis. They had about an 88% match rate in the software. And they were utilizing a software called I3S, which is a program that digitizes spot patterns and compares the number of spots, shape, size, and position within a reference area. Local research efforts include a lab up at Florida Atlantic University that is headed by Matt Ajemian. He is one of the spotted eagle ray guys, and he currently has at least one PhD student who's looking at the diet of eagle rays throughout the Gulf of Mexico to be able to really understand and compare the differences between the different life stages and where they're found throughout their life stages. Additionally, there is a large international partnership involving Mote Marine Lab, the California Academy of Sciences, and university partners from both Mexico and Cuba to understand spotted eagle rays in the Gulf of Mexico and Caribbean. And this is a screenshot from the Spotted Eagle Ray Reporting Forum, which encourages citizen participants to submit sightings of spotted eagle rays, specifically sightings that include photographs if possible. And they're using that as a loose data set that they can turn to help them continue and focus efforts and perhaps request funding. And it's a great opportunity for people to be on the lookout and contribute to this localized data set. Now, before I leave you for the day, I just want to share with you one more thing, and I'm going to have you take a virtual dive with me to a site where we regularly see spotted eagle rays. This is an artificial reef. This is the wreck of the Blue Fire, which is up off of Biscayne National Park. This is in about 130 feet of water. And what you're about to see has not been modified. This is not in slow motion. We were on our dive and we were joined by a group of 4 spotted eagle rays that circled us and stayed with us throughout. And it was really captivating because sometimes they see us and just keep going. And other times we're extra lucky to have an experience like this in which they let us stay in their world and hang out with them. And I just really encourage you to get out in the water. And if you're not interested in getting in the water, that is just fine. Go walk on the Bear Cut Bridge between Crandon Marina and UM Rasmus. And almost every day I walk out there and see a spotted eagle ray. And it just absolutely makes my day, and it's magical every time. And with that, I'd like to thank you all very much. And Ed, I'm going to ask you to put up one more poll. This is not a quiz. This is simply for me to get a feel for how this went. So, oh, I see Ed wrote me. No problem. I will launch this last poll. So if you all could just please indicate your answers. The first is simply asking whether or not you learned something from watching this webinar. I'm asking for where in the world are you? And how many people are watching this webinar? So really easy, and I thank you all again so very much. And if you're done with your responses, please go ahead and type your questions into the chat box, and Ed will be happily moderating that and answering questions.
Yep, and it's now 5:30, so if you do need to leave, that's fine. But if you do have any questions for Anna, please go ahead and type them in. And again, if you do want to receive an email every week to let you know what the topic is, you can also put your email in there and we can add you to that.
And your email will not be shared with anyone else. It's simply used to send you the topics and the registration links. I see there was some confusion about site fidelity. I hope that I did not say wife fidelity. Site fidelity simply means where a group of animals show particular interest or enjoy one area in particular. That's the easiest way to explain it. But maybe they have, you know, wife fidelity things going on too.
All right. Anna, I did have a question for you actually. Are there any local conservation organizations that we and the participants can check out to learn more about, you know, conservation efforts here in Florida?
In terms of local Miami-based, at this point, no. The closest thing to us is Mote Marine Lab, which is located in Sarasota. And I've actually reached out to them and I'm still waiting to get a response just to see what capacity they have or what need that they have for any sort of similar data, or whether or not it would be feasible to try and start a similar photo project on this coast. So nothing local, but Mote Marine Lab is definitely the closest organization that's studying this species.
Okay, great. No questions just yet. But lots of love for eagle rays.
Yes. All right. Wonderful. Well, folks, if there are no questions, please have a wonderful and safe holiday weekend. And we look forward to working with you again next weekend. I just saw Megan.
Oh, yeah.
Have I personally ever seen a 17-foot eagle ray? No, I have not. But remembering that the 17 feet is total length, so from the tip of the snout all the way, including that tail. So no, I've not seen one that big. Probably the largest one I've ever seen, Megan, is, I would have to say, 10 or 11 feet, and that's including the tail, which is usually 2 to 3 times the body length. All right. Well, guys, I thank you so much.
Thanks, everyone.
We're going to close this out and we hope to see you next week. Take care.
Thanks,
now.
Anna. Thanks, everybody.
Have a great weekend.
Thanks. Bye.